Good morning. I'm Peter Hogg. I'm a PhD student in the Haas lab. Today I'm going to be talking about an open source software project called DynaROI, which is a dynamic region of interest management software that I started developing a few months ago. And the talk's titled Universal Segmentation of Time Series Regions of Interest for Aligning Neural Activity to Morphology. So in our lab, we use two-photon microscopy. It's a type of fluorescent microscopy that is uniquely has advantages for in vivo imaging, allowing you to use fluorescent biosensors to measure neuronal activity within living animals to see better how neurons compute information by measuring proxies for neural activity like calcium levels using genetically encoded biosensors like GCAMP, or using glue sniffer to measure neurotransmitter release across a neuron. But traditional forms of two-photon microscopy is very slow because it uses a technique of raster scanning. And this is where you use a two-photon laser to scan every single pixel in the imaging space, and then you lose a lot of time by dwelling the laser into areas where the region you're imaging doesn't actually have any sample. So you may only have one neuron labeled, but you actually spend a lot of time dwelling the laser beam into dark space where you'd get no fluorescent photons from your sample. Because this is a very slow process and neural activity is very fast, there's a lot of advances in two-photon microscopy that are designed to speed up this process. These include techniques called scan-lined angular projection microscopy, random access multiphoton microscopy, and two-photon holography. These techniques have a shared commonality that instead of sampling every single point in space with your laser beam, you actually use sample information to restrict where you excite your sample, and then only image those areas of interest. So for example, in holography, you can shape the wavefront of your laser and then use beamlets to only excite areas of interest in a volume that are predetermined. Or with scan-lined angular projection microscopy, you can have predetermined regions of interest, which you then scan a laser beam opposed to a point, and then you can get the fluorescent changes of those regions of interest very, very quickly. So I'll use an example from our lab where we use random access multiphoton microscopy, kind of how this works. You first acquire a slow referenced image volume, and then you find, for this volume contains a neuron. Then you select points of interest across the neuron's arbor that you want to measure, and then you use the random access multiphoton aspect of the microscope to only sample from those areas. The problem, though, is that these techniques often require user-defined positions or user-generated ROIs. And creating regions of interest is very slow, especially if you have a very complex structure that you're trying to predetermine areas to image from. So, and this can also lead to inconsistencies between users, because different users will have kind of biases in how they construct their ROIs. And also it makes it extremely difficult to image dynamic structures. So for example, a single brain cell early on in development, like the ones that we image in our lab, they have massive amounts of structural change. So this is a time-lapse image of a single neuron labeled with GFP, and the time interval is 5 minutes between volumes. You can see within minutes that parts of the arbor have extended, retracted, there's new protrusions occurring. And if you want to measure the neuronal activity in these dynamic structures, you have to be updating your regions of interest very, very quickly to use these fast forms of two-photon microscopy to get the neuronal activity. So the solution is to automatically generate ROIs during an image session, and kind of to remove the human from the process of generating ROIs, and then to automatically register and update the ROIs over time during the imaging session. And then you can incorporate some post-hoc analysis about the spatial relationships between the ROIs. And that's where my open source software project kind of is trying to meet these solutions. So I started work on DynaROI, which has a pipeline where you take your reference image that you acquire using the slow raster scanning technique. You then perform pixel classification, so that the pixels within your reference volume can then be put into discrete semantic categories. Then you can generate ROIs automatically based off of rules pertaining uniquely to each semantic category. And then you can repeat this throughout an experiment. You then use the ROIs during the experiment for the faster types of two-photon microscopy, either random access multiphoton or scanline angular projection microscopy. So the project's still in its infancy. I have some preliminary data where I've been able to successfully use a bit of a pixel classifier to determine what is and isn't neuronal. So I've been able to exclude a lot of autofluorescence, things like melanocytes within our imaging areas that would otherwise you wouldn't want to image, and then to begin to generate regions of interest across an entire arbor. So the goal is to pretty much be able to create a very complicated structure within minutes, and it's very, very challenging to do that. So we're currently trying to replace the pixel classifier with a machine learning based approach instead of an old school learning approach. I've rewritten the code for the ROI generation to improve the quality of that. I'm increasing the generalization of the pipeline so other labs can then use DynaROI to image their samples. I'm migrating the code to a Napari plugin to make it a little bit more user-friendly with a user interface based off of that environment. And then we're providing direct integration with ScanImage and Micromanager, so the ROIs generated can then be fed directly into microscope control softwares. So with that, I want to be able to thank my lab for their support for the project, my project supervisors, Kurt Haas and Kasper Pokorski, and the funding agencies, and particularly the Canadian Open Neuroscience Platform. So, yeah.